Hi guys, welcome to Heterotrophic Protists, the Mycota, and the slime molds. This week your reading is the very end section of chapter 15, just a bit about heterotrophs. That's all you need to read this week. And I told you we'd start talking about learning objectives. So for this chapter and for this lecture, I want you to know who the major lineages of heterotrophic protists are, how they eat, what their life cycle is like, and do you know an example of one? Like, where do they live? What kind of habitat would you find them in? And what are some common examples of these creatures? It's just very general things, just because I want you to get introduced to them. And they are funky. These are funky organisms. The kind of organisms you just don't have a clue of how they fit into the tree of life. And it's not a crazy idea because these things have been moved around the tree of life several times in my lifetime. When I was in university, these things were considered fungi, but they're not. They're distantly related to fungi. They just share some traits with fungi, but phylogenetically and molecular evidence shows that they are very distinct. Well, some more distinct than others. They're beautiful though, like everything I think in the world has a special kind of beauty. This beauty is hard to see sometimes because they're not all, even when they're present in an environment, you don't always see them. You only see them when they come out to fruit. And you only see the structures, the fruiting structures, as distinct and deliberate structures when you get up really close. If you look at them from a distance, it just looks like ooze or slime or dog vomit. There's two videos I've posted this week. One's called Are You Smarter Than a Slime Mold? And the other one is some time-lapse photography. It just shows you how incredibly bizarre these creatures are. So please check them out. So in general, taking a step back, when we think about protists, all protists, they're divided into three groups based on their nutritional habits, how they get food. First group could be thought of as more animal-like. And these protists are heterotrophs. They phagocytize things. They're like amoeba and paramecium, right? They're hunters. They go around looking for little things to eat, and they eat them. The second group, they're more plant-like, and these are the protists that photosynthesize. And we've already covered these ones a little bit. They're the autotrophs. The third group are the protists that are more fungal-like, and they're the decomposers. They have absorptive nutrition. They can even have things that look like hyphae. They're not fungi. They just have the same kind of habits as fungi. And here you can see slime molds and Phytophthora growing on a potato. These are the ones we're going to talk about today, the heterotrophs. And they're kind of fungus-like, and like I said, they were long considered to be fungi. But they're not fungi. They're in completely different supergroups. Now, this is a first class where I want you to start thinking about names, specifically supergroups. Which supergroups do these organisms belong to? And what are class names? So the supergroups are Straminopola. The Mycota belong to Straminopola, and the Myxomycota and the Dictyosteliomycota, they belong to the amoebozoa. So let's take a look at this drawing from your text, which I want you to know. By the end of the semester, I want you to be able to know something about every single one of these supergroups and the creatures that we talk about within them. So today we're talking about the oomycetes, and they're all the way up there in Straminopola. And we're talking about the slime molds, which have their own supergroup, the amoebozoa. Now, Straminopola, let's stop there because you guys are probably familiar with Straminopola. These are the macro and some micro algae. These are big. They can be big and they can be little, but you've probably seen them. You've seen diatoms, you've seen bulk kelp, you've seen all of these things. These are the algae we're really familiar with, brown and golden algae and diatoms. And Phytophthora and oomycotans. And they look kind of like this. They're mostly unicellular. They live in the water. The reason, other than molecular evidence, that puts them in with the Straminopola is that they have this characteristic two flagella zoospore. They're called zoospores, not because they're animals, but because they can move. But they're just asexual spores, and this type of spore is conserved within the Stramonopula. So there's molecular and there's morphological evidence that puts these strange creatures in with the Stramonopula. But the Mycota look very different than the other Stramonopula. They look like fungi. If somebody showed me this picture, I would have said for sure it's a fungus. It looks like a hypha with sporangia. It's not. Primarily because the cell wall is cellulose. It's not made out of chitin like fungi. And these creatures, they live in the water. They live in the water, and like fungi, they're plant pathogens, but there's not very many of them. One of the most famous types is Phytophthora, and maybe you guys have heard of this. This is a really huge global concern right now in North America, not as much in Canada, but in the United States and down south and in Australia. It's ravaging forests, like completely taking forests out. This is Phytophthora. I'll show you some more examples of Phytophthora in a second, because it's by far the most famous Mycotan. But let's talk about the life cycle. Now, this is a figure from your text. And remember in one of the first lectures, I said, I want you to know life cycles, zygotic, gametic, or sporic. Remember the cycling between haploid and diploid generations, between unicellularity and multicellularity. Gets hard to figure out where these organisms fit in because their structures are very different and they have these complicated asexual phases added onto their life. However, if you ask yourself a couple of questions, you can usually figure it out. First of all, where is meiosis happening? Well, there it is. Meiosis is happening right there in the oogonium and the antheridium, which answers another question. These guys have gametangia. Where's the multicellular stage? Well, this is the multicellular stage. It's got a multicellular diploid phase with gametangia, and meiosis happens in the oogonium, in the antheridium, to produce these haploid gametes. These gametes then fuse to produce a zygote. The zygote is not multicellular, and on and on and on. So this type of life cycle is gametic. Structures look a bit different, but it's pretty much well conserved there. So it's got gametic meiosis, and you can see here the oogonium. It's got a giant oospore. It looks like a fungus. I swear to God, these things look like fungi, but they're not. Only the sporophyte is multicellular. It has gametangia. Like I said, let's look at that a little bit more closely. We've got the antheridium. Now, if you hear antheridium in this class and in other class, it's a term used in botany and in microbiology, I guess, to connote specialized male reproductive organs. Specifically, they produce the spores produced in the antheridium or sperm cells. In plants, these guys don't have sperm cells, so they're called gametes. Let's just call them gametes. The oogonium is where you're going to find the egg cells, which go on to become the oospores. So the zygote is the oospore, and that goes on to continue the life cycle. So what about this Phytophthora? I said I'd come back to it. Well, here's a picture of a tree in probably in California that has Phytophthora. It does not look like, it looks like it's bleeding, the poor thing. Phytophthora ramorum is responsible for killing millions and millions of trees since the 1990s when it was first noticed or introduced in California and Oregon. Don't really know where it's come from, but it's just done a job on oaks and other trees. It can affect other trees as well. The reason Phytophthora is so pernicious is because it does really well in wet and dry conditions, because it's got two different kinds of spores, the sexual spores and the asexual spores, and they're both adapted differently. So the zoospores, the sexual spores, are spread when it's wet, and remember those little flagella, they can swim and they love wet soil. So the zoospores are the asexual. The oospores, the sexual spores, are more resistant. They've got these really thick wall coverings, and they can just sit there and wait for decades, maybe years, for sure years, until conditions pick up or more hosts come along. And this is a cross section of a leaf. You can see the epidermal cells and the epidermis, and the zoospores germinate with their two little flagella, and they grow into the palisade parenchyma, and they go to town and kill the plant. Eventually they get into the xylem and phloem. Okay, now this organism was responsible for the Great Famine in Ireland. You guys have probably heard of this. Maybe you have relatives. If you've gone to Boston or Dublin and you've seen pictures of emaciated people because so many people starved to death. The population of Ireland was reduced by a third. 1 million people died. 2 million people immigrated. My relatives immigrated during the Irish potato famine, which is called the potato famine because Phytophthora attacked the potato crop. Now it's kind of funny, or it's not funny, haha, but it's strange. Potatoes were not introduced to Ireland until almost 1600, and it was Raleigh, Sir Walter Raleigh, who introduced them, right? Because potatoes are a South American and Central American crop. They originated in the Andes somewhere thousands of years ago, but it wasn't until Europeans started colonizing and exploring the rest of the world that they brought these crops back to Ireland. So in 200 years, potatoes became the only crop in Ireland, which seems hard to understand how that could happen. So potatoes came from South America, right down here near Peru. But so did Phytophthora infestans, and the Phytophthora infestans is the Phytophthora that attacked the potatoes. And it in the 1880s. So potatoes went to Ireland in the 1600s. It wasn't until the late 1800s and the early 1900s that it made its way to Europe and around the world. And boy, did it make its way to Ireland. Conditions were perfect for an infestation of Phytophthora because the Irish were monocropping one variety of potato which had no resistance to Phytophthora. So in a very short period of time, their entire fields just rotted, and there's nothing they could eat. Well, this is an example of a potato affected by Phytophthora. Why did it happen? I used to think that it was the farmer's fault, right? What are they doing growing one crop of potato? We know that's bad news now. And they did too. The farmers, people have known this for a long time, right? You have to have diversity in seasons and genotypes, even if they didn't know they had genotypes. But it wasn't their fault. Because what happened, now when we think of the United Kingdom today, right? At least the way it looks today, it might change, as there's Scotland, England, Wales, and Northern Ireland. But back then, Ireland was colonized. There was no Northern Ireland. It was colonized by the British. And England and France were busy carving up the planet, claiming different parts of the world. The armies were busy colonizing. And colonialism needs, you got to feed those soldiers. So Ireland was being forced to produce food for the British army, and to the expense of itself. People in Ireland were not able to feed themselves. And in many districts, their only food is potato and their only beverage water. That's what it was like in Ireland before the potato famine. And then Phytophthora hit, and whew. So just a little backstory, another little story about the evils of colonialism. Because you hear about the story of the Irish potato famine, but you don't realize, at least I didn't, that it has a dark colonialist side to it. Okay, so that's all I want you to know about Mycota, right? Phytophthora, the life cycle, water molds, that kind of thing. Myxomycota. Myxomycota is an amoeba. It's called an acellular slime mold. Acellular because it's just like a giant cell with many nuclei. So it's also called cenocytic, this growth habit, when you've got this body that instead of being made up of many cells, it's just a continuous body with no cell division, and the nuclei are just circulating back and forth. It's other creatures behave this way, but it is the exception. So it's a bit of a weird way to live. So don't remember what was in that little box, but I bet we'll find out soon. What is the life cycle like? I mean, superficially, it's super easy. You've got this diploid phase, which is like this acellular, tons of nuclei in this big ooze that forms a sporangium. The whole ooze forms itself into sporangium and divides meiotically to form all these spores, these haploid spores. These spores then grow into amoebas, and they either fuse their nuclei, karyogamy, or they fuse their cytoplasms and continue as this dikaryon phase with one cytoplasm with two nuclei for a little while until eventually karyogamy. And you get a 2N zygote, which then reproduces the nuclei only to have this big nuclear, multinucleate plasmodium. So what life cycle is that? Well, this is zygotic, gametic, sporic. One of the hallmarks of delineating these lifestyles is that we're cycling between single and multicellularity. These things aren't really multicellularity. The plasmodium is one cell. It could be giant. It could be like 10 meters squared, but it's one cell. It's not really, it doesn't really fit into these categories. But the cool thing about this, and this is, we haven't talked about this yet in this class, but it's not uncommon among fungi, is this idea of plasmogamy, where it's not karyogamy. It's not nuclear fusion, but it's protoplast fusion. And karyogamy is nuclear fusion, but plasmogamy, some organisms spend a long, long time in the state of plasmogamy with two nuclei just paired in a cell without fusion until something triggers it to fuse. Sometimes it's never triggered to fuse, and it'll continuous life cycle as a dikaryon. Okay. So for these creatures, see, here's the sporangia. These guys are so beautiful, and if you just see them from a distance, they look like ooze, and you get up close and you see the intricate detail of these sporangia. They keep growing as this plasmodium as long as there's fluid and moisture, but as soon as it's dries up, it sporulates, and the protoplasm just splits into separate spores. It's nothing really complicated. It's pretty easy. And then when times improve, the spores split open and the protoplast gets back to eating. Really kind of simple organism, but it's so beautiful. Now, sometimes when conditions are rough, like really quickly, it doesn't have time to sporulate, becomes a sclerotium. And a sclerotium, this is what really looks like dog vomit. Sometimes you see this on the forest floor. And it just dries up. It just insists. It dries up. And when it gets wet again, it'll start growing again. So kind of simple organisms. So that was Myxomycota. In the same group of amoebozoa, we have the Dictyosteliomycota. And these are the cellular slime molds. So the last ones we saw were the acellular slime molds because they were just a plasmodium. These are cellular, so they have distinct cells. And they're very beautiful, and they look and behave very similar to what we would consider a typical amoeba. They live in the soil, they're free living, and they feed on bacteria. So I said that these are fungal-like because they have hyphae and absorptive nutrition. Well, these guys are actually hunters, and they are predators. They eat bacteria. But some fungi are predators too, so there's always an exception. So these myxamoebas, they're called myxamoebas. They just reproduce by cell division. They divide, they divide, they divide until they run out of food. When they run out of food, these little amoebas aggregate together and form a giant slug. They're so beautiful. I mean, seriously, when You've got to see videos of this. I didn't put a video on, but I should. Just a coordinated orchestration of these single-celled organisms, and they do this most beautiful dance. They chug along as a slug-like thing, and it could be 100,000 individuals, right? So there's 100,000 cells in that. It's called a pseudoplasmodium because they're individuals. They're not one body. They're individuals cooperating. So it's not a true plasmodium. And here you can see there's a micrograph of these little individual mixed amoebas lining up, getting ready to make a slug. And when they are slug, they chug along, they move to a new habitat where conditions are better, and then they release spores. Now, these guys can move pretty fast, 1.5 millimeters per second. When you're as tiny as that, I think that's pretty incredible. So here's what happens. So they chug along as a slug. They get to this new place, and they start differentiating. Remember, there's 100,000 individuals in the slug, and they differentiate. They all do, within 24 hours, they produce a fruiting body. And it's like this, it's like the circus. They all get together and make this, some guys aren't, it's like, at the circus, they build these pyramids with some people on the top, some people on the bottom. That's what they're doing here, except for the people on the top become spores. So the stalk cells, so the cells near the front of the slug became the stalk, and they die via apoptosis. So they just program cell death. Spore cells are the individuals near the rear of the slug. They differentiate into spores. So it's like it's an organism, but it's not. It's cooperation among organisms. Each spore then releases a single mixed amoeba, and the whole cycle starts again. These little amoeba-like creatures. Well, who gets to be a spore and who gets to be a stalk? It's programmed. It's genetically programmed. There's a gene that enforces cooperation among cells. When it's absent, the cells keep on climbing to the top of the stack, but when it's present, they stay on the bottom and differentiate. So wild. So their life cycle, it's so not that hard. I mean, it's weird. It's weird. You can't really categorize it because, again, it's a single-celled organism. It becomes cooperative, but it's still single-celled. So let me walk you through it. So you've got this vegetative cycle where the mixed amoeba just split. As long as conditions are going, hunting and is good, they just keep on going. But sometimes they have sex where the haploid amoeba will fuse, have a plasma gamete with these two nuclei in it, and it becomes a voracious predator at this point and starts eating all of the other mixed amoeba until they're all gone, and at which point this giant diploid creature becomes a macrocyst, and this macrocyst undergoes meiosis, and we've got the mixed amoeba again, which I just think it's hilarious how voracious the zygote gets and just gobbles up all the other mixed amoeba. Okay, so you've got the vegetative cycle, you've got the sexual cycle, and then you've got the social cycle, right? When conditions are bad, it has vegetative reproduction where it forms these slugs in the fruiting bodies, right? So there's two types of vegetative reproduction. There's times are good and times are bad. So it's complicated. These life cycles are complicated. The simple organisms with complicated life cycles. Okay, that's all I want you to know. So the questions for this week, which could be on a final or a version could be on a finals, who are the heterotrophic protists? Tell me what you know about them. Which ones are plant-like? Which ones are animal-like? Which ones are fungal-like? Describe the cycles and growth habits of each one. All right, that's all for today. I'll talk to you soon.